Hi guys, welcome to the tutorial section of the El-Igo channel. I'm George Omogbeme for the El-Igo channels. On today's tutorial, we are going to be looking at ABO and Rhesus blood grouping. Alright, and the technique or the method we are going to be using is called the tie technique or the tie method. And the type of grouping we are going to be doing is called the cell grouping or forward grouping. So on our tutorial today, we are going to be looking at how to determine the blood group of an individual or the blood group of a given sample. So on today's tutorial, I have two samples right here. We are going to determine the blood groups, and of course, if you look right before me is my Antisera. Okay, I have my Antisera A, Antisera B, Antisera AB, and of course Antisera D. So quickly, without wasting time, all we have to do is to take from each of the samples, all right, and spot them in four different places. Then I add my Antisera. This is the Antisera A. Just a drop. We use this to determine the rhesus factor of the blood group. I will just mix. Okay. I'll mix the Antisera and my cell together. All right. This to allow for proper reaction. So after that, just mix this a bit, then swirl anti-clockwise motion, and you can see that the reaction is already visible. Okay, the reaction is already visible. Okay, and of course, in no time, our blood group is ready. So if you look very well, you will see that there's agglutination reaction in the anti-D column. Okay, and of course there's no reaction in every other column. So what this means is that this sample is O rhesus D positive. That's the blood group of this sample is O rhesus D positive. All right, and of course the second one, if you look very well, you see that on the A Antisera A part there is agglutination reaction. Then of course the Antisera B there is none. And at the Antisera AB there's of course reaction as well. At the D part there's also what? Reaction. All right. So what this means is that this sample is A rhesus D positive. A rhesus D positive. Using this Antisera, one can determine the blood group of various types from different individuals. I would like us to note these three important techniques when carrying out our blood grouping. One, you can actually put drop of your Antisera on the ties before adding drops of your red cell suspension. Alright, this is to help prevent contaminating the Antisera by way of allowing the tip of the dropper to touch the cells that is already on the ties. Two, ensure to always use 20 to 30% red cell suspension. Alright, this will help reflect the color of your Antisera on your ties. This can help prevent or detect errors. Okay, on this tutorial video, I use 100% red cell suspension so that the reaction can be visible on camera. And 3, mix from right to left. All right, because the avidity of O cells is usually longer than that of A cells and B cells. And also, in the case of presence of weak antigen, formerly known as DU antigen, as this will save time. So also the Antisera AB can serve as your control, because whenever there's reaction on either the A cell or B cell column, there is always a reaction on the AB cell column. All right, so practice, practice, practice. See you on the next one, guys. Godspeed.